Hello friends, this is Rashmi Singh and welcome to my channel Unraveling Science. Class 9, Chapter 5, The Fundamental Units of Life, Cell. Introduction. Cell is a structural and functional unit of life. All living organisms are made up of cells. They either exist as a single cell or as a combination of multiple cells. Discoveries about cell. Robert Hooke in 1665 noticed the presence of cells in the cork slice. Leeuwenhoek in 1674 found the presence of living cells in the pond water. Robert Brown in 1831 recognized the existence of nucleus in the cell. Purkinje in 1839 invented the term protoplasm, which is a liquid present in the cell. Sledeen and Schwann in 1838 and 1839 presented the cell theory that all organisms are actually made up of cells. Virchow in 1855 suggested that all cells come from the cells that already exist in nature. The cell theory. According to cell theories, all the plant and animals are composed of cell, and cell is the basic unit of life. All cells arise from pre-existing cells. Types of organisms. First is unicellular organism, which are the organisms that consist of single cells such as amoeba. Second is multicellular organisms, which are the organism that contains various cells that perform different functions in the organism, such as fungi, plants and animals. The shape of the cell. The shape of the cell may vary depending upon the type of function they perform in an organism. Cells are capable of changing their shape. For example, white blood cells and amoeba can change shape on their own. Structure and components of a cell. A cell contains three features. First is plasma membrane, second is nucleus, and third is cytoplasm. First, let's know about the plasma membrane. You can see here in the picture, it is the outermost covering of the cell that separates the contents of the cell from its external environment. It allows or permits the entry and exit of some material in and out of the cell. That is why it is also called as selectively permeable membrane. It is made up of lipids and protein. Cell wall. Cell wall is another rigid outer covering in addition to the plasma membrane found in plant cell. The cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose, and cellulose is a complex substance which provides structural strength to the plant. Cell wall permits the cells of the cell to withstand very dilute hypotonic external media without bursting. How can substance move in and out of the cell? Gaseous exchange between the cell and its environments takes place through diffusion. What is diffusion? It is a process in which gaseous substance move from higher concentration to lower concentration. Movement of water between the cell and its external environment takes place by means of osmosis. Osmosis. It is a process in which water moves from higher concentration to the region of lower concentration through a semi-permeable membrane. Hypotonic solutions. As you can see here, it is if the concentration of water outside the cell is higher than the concentration of water inside the cell, then The cell will gain water by osmosis and likely to swell up. Isotonic solutions. If the cell is kept in a media that has same concentration of water on both inside and outside the cell, in this case, water will cross the cell membrane in both the directions, so the cell will remain the same size. Hypertonic solutions. If the cell is kept in an environment that has lower concentration of water than what is present inside the cell, in this case, the water moves out of the cell. As a result, the cell will shrink. Endocytosis. It is a process by which plasma membrane engulfs food and other materials inside the cell. As you can see here in this picture, amoeba acquires food through this process. Plasmolysis. When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. This phenomena is known as plasmolysis. There is a question: how plants, fungi and bacteria can exist in a hypotonic medium? Answer is: plant, fungi, bacteria exist in such situation because of their rigid cell membrane. Even if the cell swells, the cell membrane is able to prevent them from bursting out. Nucleus. Nucleus is the prominent organelle present in the cell, which is the controlling center of all the activities of the cell. Nucleus has a nuclear membrane that covers it all around. Then there are pores present on these membrane that allows the movement of substances in and out of the nucleus. There are chromosomes, rod-shaped structures present in the nucleus which contains genetic information. The chromosomes contain two types of things. First is DNA, which is responsible for organizing and constructing new cells. And second is protein. These help in packaging and condensation of DNA. Chromatin. As you can see here, these are thread-like material present in the cell. The chromatin organizes itself into chromosomes whenever the cell is about to divide. Nucleolus. It is called the brain of the nucleus. It comprises 25% of the volume of the nucleus. It consists of protein and ribonucleic acid, which is RNA, and helps in the formation of ribosomes, which are responsible for formation of protein inside the cell. Nucleoid. Sometimes cells do not have a well-defined nucleus due to the lack of nuclear membrane. Such nucleus with no definite nuclear boundaries is called a nucleoid. Difference between prokaryotes and eukaryotes. Prokaryotes, the cell of organisms lack nuclear membrane. In eukaryotes, cell of organism have nuclear membrane. In prokaryotes, nucleolus is absent. In eukaryotes, nucleolus is present. Single cell chromosomes are present in prokaryotes and single or multiple chromosomes are present in eukaryotes. Reproduction is always asexual in prokaryotes, while reproduction is both sexual and asexual in eukaryotes. Prokaryotes are always unicellular, while eukaryotes are often multicellular. Membrane-bound cell organelles are absent in prokaryotes. Membrane-bound cell organelles are present in eukaryotes. Centriole is absent in prokaryote. Centriole is present in eukaryote only in animal cell. Cell division is by binary fission in prokaryotes. In eukaryotes, cell division is by mitosis and meiosis. Examples of prokaryotes are bacteria and blue-green algae, etc. Eukaryote examples are fungi, plant cell and animal cell. You can see the picture of eukaryotes and prokaryotes over here. Now let's come to cytoplasm. The cytoplasm is the fluid content inside the plasma membrane. It is also contain many specialized cell organelles. Each of these organelles perform a specific function for the cell. Functions of cytoplasm. It helps in exchange of material between cell organelles. It acts as a store of vital chemicals such as amino acid, glucose, vitamins and iron, etc. It is the site of certain metabolic pathways such as glycolysis. It dissolves cellular waste. Endoplasmic reticulum, also called as ER. The endoplasmic reticulum is a large network of membrane-bound tubes and sheets. The structure of ER is similar to that of the plasma membrane. Two types of ER are there, rough ER, you can see here in this picture, and the smooth ER. So the rough ER contains ribosomes that are responsible for manufacturing of proteins in the cell. They give us rough texture to the cell. While smooth ER manufactures fats and lipids in the cell which allows the functioning of the cell. Now functions of ER. Transportation of material between different parts of the cytoplasm and also between nucleus and cytoplasm. Second is folding of proteins which are synthesized by ribosomes on rough ER. Then detoxifying poisons and drug out of the cell. Golgi apparatus. Now Golgi apparatus, you can see over here the picture of it in the cell. Camillo Golgi discovered the Golgi apparatus. The Golgi apparatus consists of a system of membrane-bound vesicles arranged approximately parallel to each other in stacks called cisterns. These membranes often have connections with the membranes of ER and therefore constitute another portion of a complex cellular membrane system. Functions of Golgi apparatus are: it carries material synthesized by ER to different parts of the cell. The material is stored and packaged in vesicles. Formation of complex sugar. Formation of lysosomes. Lysosomes. Lysosomes are the kind of waste disposal system of the cell. It helps to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Lysosomes have membrane-bound structure whose sacs are filled with digestive enzymes. Functions of lysosomes are: lysosomes can break down macromolecules, old cell parts and food and foreign invaders like bacteria. During the disturbance in cellular metabolism, such as when the cell gets damaged, lysosomes may burst and then digest their own cell, therefore they are known as suicidal bags. Next is mitochondria. You can see over here the mitochondria in the cell. It is also known as the powerhouse of the cell. It is a double membrane organelle and has its own DNA. This is the picture of a mitochondria. The outer membrane is very porous while the inner membrane is deeply folded. These folds create a large surface area for ATP generating chemical reactions. Functions of this mitochondria is: the energy required for various chemical activities needed for life is released by mitochondria in the form of ATP, which is adenosine triphosphate molecules. ATP is known as the energy currency of the cell, and the body uses this energy stored in ATP for making new chemical compounds and for mechanical work. Mitochondria have their own DNA and ribosomes, therefore mitochondria are able to make some of their own proteins. Next is plastids. Just like mitochondria, plastids are also double membrane organelles and they have their own DNA and ribosomes. Plastids exist in plant cell only. Depending upon the type of functions they play in the cell, they are classified as following. You can see here different types are chromoplast, chloroplast, leucoplast, etc. First is chromoplast. They are colored in nature and contain pigments called chlorophyll, which causes photosynthesis in plants. They contain orange and yellow pigment and can be further divided into chloroplast. Next is leucoplast. They are colorless in nature and acts as a storage space of the cell. They contain starch, protein and oil. They can be further divided into amyloplast, elaioplast and proteinoplast. Amyloplast. They are found in tubers, cotyledons and endosperm of the plant. They are used to store starch. Elaioplast. They are found in epidermal cells of the plant and they store oil. Proteinoplast. They are found in seeds and nuts and they store protein. Chloroplast. Now we will study the chloroplast in detail. You can see the picture here. Chloroplasts contain a photosynthetic pigment, chlorophyll, along with lipids, carbohydrate, mineral, DNA, RNA, grana, thylakoids and stroma. The main functions of chloroplast are conducting photosynthesis in plants, protein synthesis, release of oxygen and storage of starch. Vacuoles. Vacuoles are the storage sac for solid and liquid contents. In plant cell, vacuoles are large and present in the center of the cell. In animal cell, vacuoles are small and scattered throughout the cell. In plant cell, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. Many important substances like amino acid, sugar, various organic acid and proteins are stored in vacuoles. In animal cell, vacuoles contain food items in unicellular organism like amoeba. Vacuoles help in expelling excess water and waste products from the cell. You can see over here the plant cell have large vacuoles and the animal cell have multiple small vacuoles. For difference between plant cell and animal cell, you can visit my channel. There search for playlist of class 8th and you will find this video on plant and animal cell differences. Thanks for watching. Please do like, share and subscribe.